Thank you, Sid, for the kind introduction. So my name is Kedar and I'm a PhD student. I have just started in January, so it's just 3 months now, and working as a Marie Curie doctoral candidate here at the University of Manchester. So I'll be talking about how do we try to understand the brain in case of Kabuki syndrome at a cellular level. So first of all, there is no any medical treatment available for Kabuki syndrome, as we all know. For internal organ malformations, there is surgical correction available. For systemic problems, there are regular medical follow-ups available. But for developmental delay or brain-related disorders, there is only supportive care available. So because of that, there is a need to understand how the brain in Kabuki syndrome is functioning. So, but brains, to access the brains in the patients, it's difficult and it's impossible. So for that, we have alternative approaches like cell-based models or animal-based models. So here we are taking a cell-based approach to understand the brain, and specific in the cell-based approach, we are using stem cells. So what are these stem cells? So we take patient biopsies, that's the skin biopsies. We take skin cells from the patients, which are called fibroblasts, or even we can take blood cells from the patients. Then we provide them with certain factors. These are called reprogramming factors, which convert these skin cells into a stem cell. And stem cell is a very special type of cell which can get converted into any type of cell in our body, including the neurons. So, because our project is more focused on the brain, we try to derive neurons from these stem cells. So there is a protocol or a procedure for converting these stem cells into a neuron. This happens through a specific stages of protocol that we follow. We provide these cells with certain factors so that they can follow a developmental path into a neuron. And specifically, we try to generate cortical neurons because that's the part in the brain that is affected mostly in case of Kabuki syndrome. And further to that, we try to do DNA, RNA, protein analysis with these neurons. And also we try to study the electrical activity that is happening in the cells of derived from these patient cells and the control cells. So as we know that these cells contain DNA and DNA contains some genes, but all the cells in our body has same DNA, but the genes that are present in that DNA are getting expressed differently at different cells. So for example, in a nerve cell, these 4 genes are on, 3 are off, but in case of muscle cells, these 3 are on, but 4 are off. So, and only the genes that are on, they get converted into a RNA. That RNA translates into a protein. So that's the kind of functioning how it happens. And because of that, we try to study the RNA so that we can map that onto which genes are on, which genes are off in case of neurons, in case of Kabuki syndrome. So for this RNA analysis, what we do is we do a comparison between a control group and a Kabuki syndrome group. And ideally, if there are no differences, we would find this plot as completely blank. But we do observe some differences while comparing Kabuki and a control group. And here you can see in this plot, these little dots, they represent a gene. And when the genes are on the right side, that means that these particular genes are getting more expressed in case of Kabuki syndrome, and these genes on the left side, they are getting less expressed in case of Kabuki syndrome. And we do this kind of analysis at all the different stages of conversion of stem cells into neurons. There are 3 specific stages that we take into consideration. That is the stem cells, the progenitor, which is the intermediate step, and the mature neurons. And at each stage of this conversion, we find some genes that are going up or going down. And we then try to find out some therapeutic approaches that can target these particular genes, which can reverse the mechanism of Kabuki syndrome and in order to have some better therapeutic strategy. So overall, what this means in a cellular context. So just to give an analogy, just imagine traffic lights. When the traffic signal is green, cars move ahead. When there's a red light, they stop there. And this coordination between red light and green light helps to move the traffic in a proper way. But in case of Kabuki syndrome, this traffic signaling system is quite disturbed, it's imbalanced. In that case, the off controller, the red signal is a little bit more than that of green signal, which is the on controller, which makes the flow of genes or the flow of traffic kind of imbalanced. And because of that, at a cellular level, we see the cells become stiff, compact, and so this is the overall picture that what we kind of get to know from our data. So this has been the work till date that we have done. So from here, we want to transition from a 2D model to a 3D model. As we know that this brain is a nice complex 3-dimensional structure, and it's difficult to study this complex 3D structure into a 2D, like a monolayer of culture, because growing cells on this flat petri dish, we kind of don't get to see many cell types that are available in this, and which makes us to go a step ahead with that and to make brain organoids. These brain organoids are nice 3D structures that we will be using ahead in our research to study the brain in Kabuki syndrome. So what are these brain organoids and why are they important to study? So if you look at a petri dish in our laboratory, these are like a 2-centimeter ball-like structures, which we call them as organoids, 3D brain organoids, and they are very important to us. If you zoom into that, like look at them under a microscope, they would look something like this. And if you see at this point here, we can see some flower-like structure, which we call them as rosettes. And if we stain them with certain antibodies specific for certain cell type, then we see a nice picture, and these different colors here represent different cell types. So essentially, we get to see many different cell types in the organoid as compared to a human brain, and we also see this nice layering pattern, which is kind of similar to what we observe in case of human brain. So that's why these 3D brain organoid systems are very nice way of studying developmental disorders and specifically Kabuki syndrome. So next question would be, how do we generate these organoids? So if you look at this video, we use certain factors to collect these stem cells. We put them in microwells where these cells have a tendency to self-aggregate, form ball-like structures, as you can see here in this video. Then we take those balls out of it, provide them with certain soup of factors, certain chemicals, and we try to make them mature over a period of days. And finally, we get a brain organoid. So this is the work that I have been doing in the last 2 months. We have generated brain organoids for 40 days, and you can see at day 30, we are able to see this rosette-like structures, which make us very happy. That yeah, because with this we can say that these organoids are becoming mature and they are good to study the disorder that we are looking at. Further to this, we will be doing some microscopy, advanced microscopy images with this. We'll do RNA analysis similar to that I discussed in my previous slides to find out what novel things that we get out of here. And so that's the future work that we are planning to do. So I'll head over again to Sid.